Still standing in the room and discussing the great talk by Harry Bühler, I would ask them either to sit down or to leave the room because we're going to start the next session. Thank you very much. Okay, so thanks a lot. I think we can start. I don't know about the lights here in this room. Can we dim them a little bit? Operator? Because I don't see anybody. I have the light right in my face. Okay, so I just start under these conditions we just have. So welcome to this session, which is the vessel wall with a focus on endothelial cells. And I'm sitting here all alone doing the moderation job, but I'm very happy to be in good company by three excellent speakers, at least three excellent presentation titles, which I look very much forward to. Some household messages. So you've probably heard it before, but it's not allowed to take pictures of the presentations. And if I see people taking pictures, I am going to repeat the request. And that's Rick van Gorp. And he will talk about the effects of the old school warfarin and the novel anticoagulants in the context of atherosclerosis. Please.
Good afternoon, everyone, and thank you, Rory, for this nice introduction. And I want to start that this project is supported by Natto Pharma, Boehringer Ingelheim, and the Trombose Stichting Nederland. So we know that we have atherosclerosis, everyone has it, we develop it, and this grows over time. What's really bad is if this ruptures, and this can then lead to thrombosis. And this together we call atherothrombose. And what we know is that the coagulation factors play a role in atherosclerosis. Here, for example, we show that thrombin can influence atherosclerosis at the early time point of early stage. For example, by influencing the cytokine expressions in endothelial cells, but also in a late stage, and this is in a vascular smooth muscle cell, by migration, proliferation, and differentiation of the vascular smooth muscle cell. Here depicted on the right. And then, of course, we can ask our question. If we give oral anticoagulation medicine, what is then the effect on atherosclerosis? We can think if we give oral anticoagulation, it affects the coagulation, and thereby we have an indirect effect on atherosclerosis. But perhaps there is also a direct effect of oral anticoagulation on atherosclerosis. And now we know from the first studies with dabigatran etaxylate, the direct thrombin inhibitor, that there indeed seems to be a beneficial effect on atherosclerosis. But besides dabigatran, we also have our old school, as Rory nicely mentioned, vitamin K antagonist. And we know these induce anticoagulation via inactivating the vitamin K-dependent proteins in the coagulation cascade. However, these vitamin K-dependent proteins are also present in our vasculature, and I want to take you directly to this protein MGP, or matrix Gla protein. We know this is a very important calcification inhibitor, so by administering vitamin K antagonist, we also inactivate MGP, and indeed already studies have shown that it increases vascular calcification. So this together made up with the following aim. And that is actually just, what is the effect of oral anticoagulation treatment on atherogenesis, but then also specifically on vascular calcification. And to study this, we used the ApoE knockout mouse model, which are prone to develop atherosclerosis. And we gave these a high-fat diet, Western-type diet, which we can see as a hamburger diet for us humans. And we supplemented this diet with either dabigatran etaxylate or the vitamin K antagonist warfarin. And I first want to show you that the diet did not affect our results, since the weight of the animals, cholesterol levels, and triglyceride levels were similar between all groups. In addition, we give anticoagulation treatment, so we also want to know, does these animals really have anticoagulation? And I hope this shows that indeed dabigatran and warfarin levels were increased upon their respective treatment. And we also see that after warfarin treatment, there's an increase in prothrombin time, indicating that there is indeed anticoagulation. So now we know the diet did not influence our results, and we know that the anticoagulation treatment really induced anticoagulation. So therefore, now we can look at what is the effect on atherogenesis. So for this, first we looked at really the development of the plaque size from 42 days towards 140 days. So in this period of time, how much did the plaque size grow? And what we can see is that upon warfarin treatment, we can really see that the plaque size increases way more as for control or dabigatran ethoxylate treatment. But of course, only plaque size is not a real, it's a nice indication, but there are more important effects. And that's, for example, by the plaque phenotype. Is this plaque also more vulnerable for rupture and thereby inducing thrombosis? So first, of course, we looked at vascular calcification. And first we did this by using a micro-CT, which is depicted on the top. And what we can see nicely, what also has been shown before, that the warfarin treatment induces more vascular calcification. And this effect is not present after dabigatran ethoxylate treatment or control. We also confirmed this micro-CT data with using alizarin red staining, which just stains the calcium deposit reds in the aortic arches. But of course, we have more markers for plaque vulnerability, and one of these markers is the elastin breaks. And we can see that also upon warfarin treatment, we have an increase in elastin breaks in comparison to dabigatran ethoxylate treatment or control, and the same effect we actually see for oxidative stress in this plaque. Of course, I mentioned in my introduction that the plaque calcification can be affected by matrix CLA protein. So therefore, we also looked in the levels of the inactivated form of MGP, or so-called uncarboxylated MGP. And what I show here is that we already see there is an increase of these inactive MGP after 6 weeks of treatment. And this is especially of interest since after this 6 weeks, we still see no calcification. This occurs on a later time point. But what we see is that there is an increase still after 20 weeks of treatment. And we also know if MGP would be active, it's also an inhibitor of osteochondrogenic differentiation by bone morphogenic proteins. So if we have more inactive MGP, there should be more differentiation to our osteochondrogenic phenotype, and therefore we looked at the levels of bone morphogenic protein 4. And indeed, in line, we can see that also here, after 20 weeks of treatment, there's an increase of BMP4 levels after warfarin treatment. Of course, we also want to see, is this specifically mediated via the vascular smooth muscle cell, since the vascular smooth muscle cells is a very important creator of this MGP in the vasculature. So first, we incubated this vascular smooth muscle cell with warfarin, and we looked at the levels of MGP. And what we can see that over time, there is more inactive MGP upon warfarin treatment. And what we also see is that this inactive MGP is accompanied by calcification in vitro of the vascular smooth muscle cell. In contrast, there is no effect of dabigatran on calcification. We showed also in vivo that we saw that there is an increased oxidative stress after warfarin treatment in vivo. And we show that this is also in vitro in the vascular smooth muscle cell. So we show this is an indication that the vascular smooth muscle cell also play a very important role in this oxidative stress in vivo. And again, no effect is there after dabigatran treatment. We know also that oxidative stress is an initiator of differentiation of the vascular smooth muscle cell towards the synthetic phenotype, and these synthetic vascular smooth muscle cells secrete extracellular vesicles, which we think is a very important player in plaque calcification, since it can already form the nidus on which the calcification takes place. And in line with these data, we see that after warfarin treatment, there's an increase in extracellular vesicle secretion upon warfarin treatment. Interestingly, we also see there is even a very small reduction after dabigatran treatment. But all these results are the direct effect of dabigatran on the vascular smooth muscle cell. And we know dabigatran should inhibit thrombin and thereby induces its anticoagulation effect. So what is the effect of thrombin on the vascular smooth muscle cell? So here we show that thrombin increases extracellular vesicle secretion in the vascular smooth muscle cells, and that can be really nicely reduced after incubating with dabigatran, even in a dose-dependent effect. So taking these all together, we can come up with the following conclusion. So first we have in the upper, we can see a contractile vascular smooth muscle cell, which should be the healthy form of vascular smooth muscle cell. Upon a vitamin K antagonist treatment, or what we did, warfarin, we can increase oxidative stress and thereby promote differentiation towards the synthetic phenotype of the vascular smooth muscle cell, which then we are in this part. Here again, we have an effect of the vitamin K antagonist by increasing inactive MGP. Thereby, we promote osteochondrogenic differentiation, we promote extracellular vesicle secretion, and this all combined, we promote calcification. So, what we also know is if we have thrombin, we have an effect on the vascular smooth muscle cells via the protease activated receptors. And from the literature, we know that this promotes proliferation and migration, which are a marker of a synthetic vascular smooth muscle cell. So also here, we go towards a synthetic vascular smooth muscle cell, and we show that there's increased extracellular vesicle secretion, and we know that these play a role in calcification. So by administering a DOAC, in this case dabigatran, we should inhibit thrombin and thereby eliminate these negative effects on the vascular smooth muscle cells and in total on the vasculature. But we are currently working on another step, and that is combining a DOAC, dabigatran ethoxylate, with vitamin K, because we believe that we can then have the most beneficial effect on the vasculature. Because by inhibiting thrombin, we then have the beneficial effect by inhibiting differentiation of the vascular smooth muscle cells and should reduce extracellular vesicle secretion. But also we make the optimum status for the vitamin K status of the vasculature and thereby have more active MGP, and thereby we reduce osteochondrogenic differentiation, extracellular vesicle secretion, and in total we should reduce plaque calcification and have a more stable plaque. And of course I didn't do all this work alone, so I want to thank also the group that helped me with all this work, and I want to thank you all for your attention.
Thank you very much, Rick. So I'm sure there are questions from the audience. Yeah, sorry, I can hardly see in the light.
Before
Henry,
you will kick in again, I'm going to save you. And Rick, very nice presentation. You showed an effect, and I can hear myself in an echo. You showed no effect of dabigatran on calcification. So you treated those cells with dabigatran because you supposed that thrombin would be present on these cells? Or was it just to see the effect of dabigatran on cells without the presence of any coagulation enzyme?
The effect of the calcification in vitro, that was really the direct effect of the dabigatran on the vascular smooth muscle cells to see if it has any effect on the calcification, which we wouldn't think it would have, but perhaps you have a very not in theory-based effect what you could find.
But on the vesicles or the release of the vesicles, you see a small effect of dabigatran on the release of vesicles, suggesting that thrombin might be indeed present in the medium or on the cell culture. Any idea where this thrombin is coming from, this minimal amount?
I wouldn't immediately know where then the thrombin would come from. You could think that it would be in the serum, but no, I wouldn't really know where then the thrombin would come from. It's also a very small decrease, so you can also question if this is a really relevant decrease. I think that if you look at the decrease that we get upon combining thrombin with dabigatran, that this says much more as the data of only dabigatran on the extracellular vesicle secretion.
Okay, thank you.
Okay, So if there are no burning questions, I would like to move on to the next speaker. Thank you, Rick. So the next speaker is Jelle Postuma, and he will be talking about targeting factor 10A in the context of atherosclerosis. So, please.
Yeah, thank you very much for this nice introduction. And I would like to thank the organizing committee for giving me the opportunity to present my latest work on the effects of direct inhibition of factor 10A on experimental atherosclerosis. So, first of all, I have to declare that I had financial support from the Maastricht University Medical Center by the Koolstra Talent Fellowship and the Dutch Heart Foundation, and the compound was provided by Biopharmaceutics. So atherosclerosis is a chronic inflammatory disease at site of the arterial wall upon infiltration of fats. And in this process, many processes are involved. So the macrophages, as you can see in here, they infiltrate, which is important, and over time a plaque is formed. And at a more advanced stage, these plaques can get more vulnerable for rupturing. And in this, the main determinants are the size of the procoagulant necrotic core and also the thickness of the fibrotic cap on top of the atherosclerosis. And when a plaque ruptures, it can induce an atherothrombotic event, such as a myocardial infarction or a stroke. Well, we already know a lot about this disease, but in the last few years, we discovered that coagulation factors, such as thrombin and 10A, can regulate a variety of processes involved in the cardiovascular system. However, for thrombin, this is investigated a lot the past few years. However, for the direct inhibition of factor 10A, this is hardly investigated. So, how does it work then? These coagulation factors, they can activate the so-called protease-activated receptor. And not surprisingly, they're present on many cells that are involved in cardiovascular diseases, such as endothelial cells, but also leukocytes and vascular smooth muscle cells. And where thrombin is the main activator of part 1, it's the 10A that's the activator of part 2. So we were interested in what the effects were when we direct inhibited factor 10A on experimental atherosclerosis. And therefore, we used the ApoE knockout mice that are genetically modified so that they spontaneously develop a lot of atherosclerosis. And we used 3 different groups because we wanted to test the effects on the early as well as on the advanced stage of atherosclerosis. And in the first group, we used mice that received a high-fat diet or a high-fat diet which was supplemented with rivaroxaban for 8 weeks. And the same strategy was used during 14 weeks in the second group. And in the third group, we wanted to investigate the effects on more advanced stage of atherosclerosis. So we used 20 mice. We fed them a high-fat diet during 14 weeks. So there was already atherosclerosis. And then 10 mice continuously received a high-fat diet, and the other 10 were switched to a high-fat diet supplemented with rivaroxaban. And after this, the mice were sacrificed, and we dissected the aortic arch and carotid artery for immunohistochemical staining. So first of all, we stained them with hematoxylin and eosin to analyze the quantity of atherosclerosis. And I have to explain this picture. So here in black is the group, the mice that received a high-fat diet with rivaroxaban from the start on until 8 weeks. And in red are the mice that received a high-fat diet only, as well as for the 14 weeks. And here in blue, this is the group that switched to a high-fat diet with rivaroxaban after 14 weeks to 20 weeks. And what we found was that already after 8 weeks, there was a significantly lower amount of atherosclerosis within the aortic arch. And this difference was even bigger after 14 weeks. But what was interesting is that this group that switched to a high-fat diet with rivaroxaban showed a significantly lower amount of atherosclerosis in the aortic arch. And the same trend was found in the carotid artery. And we measured the maximal stenosis of the carotid artery by atherosclerotic plaques. And we found that in the mice received rivaroxaban, in all groups, there was a lower amount of atherosclerosis in the carotid artery. So, treatment with rivaroxaban reduces the arthrogenesis. But there's more we're interested in, because not only the quantity, but also the composition of these plaques determine whether they're prone to rupture or not. Therefore, we analyzed the collagen amount. And on the left upper side, I show representative pictures of aortic arches stained with Sirius red to visualize the collagen. And with the arrows is the thickness of the fibrotic cap. And here we found that already after 8 weeks, the thickness of the fibrotic cap was significantly thicker than compared to the mice that received a high-fat diet only. And again, this was also after 14 weeks. And what was important that, as I mentioned before, that at a more advanced stage, it seems that these plaques all of a sudden gets more vulnerable. And that's what we see here also, that the mice, compared to 14 weeks to 20 weeks, all of a sudden this fibrotic cap gets thinner and thinner. And with rivaroxaban, we were able to prevent for the reduction in the fibrotic cap thickness, so the plaque stays as stable as it was. And the same was found on the right. It's also Sirius red staining, but then analyzed with polarized light. And that's the total the content of collagen in the plaques. And here again, already after 8 and 14 weeks, there was much more collagen in the plaques. And we were able to reverse that from 14 weeks to 20 weeks when we added rivaroxaban to the diet. And furthermore, important determinants were the inflammation and necrosis. And on the left, we stained for infiltrating macrophages in the atherosclerotic plaques. And we show that in the early phase, the infiltration of macrophages is quite high in the plaque. So it's an important process in the early phase. And with rivaroxaban, we significantly reduced the infiltration of macrophages in the plaques. And again, after 14 weeks, at the more advanced stage. And there also, when mice received rivaroxaban after 14 weeks, all of a sudden this infiltration of new macrophages significantly reduces. And the same was for the necrotic core. In purple is indicated the size of the necrotic core within the plaque. And this is quite procoagulant. So when it ruptures and these necrotic core are exposed to the blood, there's a lot of activation of the coagulation, which contributes to the atherothrombosis. And we showed that the treatment with rivaroxaban significantly reduces the necrotic core, and again was reversible in pre-existing plaques, after which the mice were exposed to rivaroxaban. This necrotic core decreases. So, looking more mechanistically in this, it's quite interesting in what really drives these processes and these changes. So first of all, we stained for factor 10, because that's the factor we inhibited, and we also wanted to know if this factor was less expressed in the plaques. And as we expected, actually, this factor 10 was much less expressed in the plaques in the mice that received rivaroxaban. But what was interesting, that the most prominent receptor for this coagulation factor, for this factor, PAR1 and PAR2, was significantly lower expressed in the plaques. So, this expression actually might say that there's some kind of downregulation due to a much less activation of this receptor in all these groups. So, coming to conclusion is that we showed that treatment with rivaroxaban, in which we direct inhibited coagulation factor 10A, reduces the onset as well as the progression of atherosclerotic plaques. And these quantitative benefits were accompanied by benefits in the composition of plaques, so that they are much less vulnerable for rupturing. And these processes are potentially mediated through a reduced activation of the protease-activated receptors. However, the exact contribution of these receptors still needs to be established in the future. So, come to the end, I would like to thank everybody for their attention, and of course everybody that helped with this research in Maastricht. It's quite a beautiful city, as you can see, so give it a try and visit the city once. Thank you very much.
Thank you. Thank you, Jelle, and also for keeping so nicely in time. So I'm sure that there are also questions. Yes? Please.
Hi,
continue.
Martien Bos from the Netherlands. Very nice talk, nice data. I may have missed it, but I was wondering how much rivaroxaban did you give to the mice, and if you measured plasma levels, if they were stable?
Yeah, before we started this research, we did a pilot study in which we tested different concentrations of rivaroxaban in the high-fat diet. And we used 1.2 milligrams per gram. And that means that all mice were within the therapeutic range of rivaroxaban in the blood. So between 100 and 300 milligrams per milliliter.
Okay, thank you.
So, I would like to ask some questions as well. So, I'm very intrigued by your factor 10 staining in the plaques. And it becomes less after you treat the mice with rivaroxaban.
Yeah.
So why do you think that is?
Yeah, there's it can be more. It can be because it's inhibited, so there's just much less factor 10. But also, it's also possible that every factor 10 is used. So you see a decline when it's activated to 10, and it's broken down again. But it's hard to say where it comes from and why it's reduced. But it's most likely because we inhibit it with rivaroxaban.
Okay. Because it doesn't really make that much sense if you add rivaroxaban that you get less factor 10, because otherwise you would actually prevent the consumption. But just that's something for a glass of wine. But actually, it's been known for a long time, and it's also something that intrigued me a lot, is that factor 10 is also a ligand for MAC1 on macrophages. So have you tried co-localizing your factor 10 with the macrophages? Because maybe it's just a side effect of the macrophages becoming less in your regressed plaques.
Mm-hmm. Yeah, we did not investigate the co-localization with factor 10, although we measured the co-localization with PAR1, and we found that PAR1 was strongly correlates with the macrophages. So still, if the effects is there, we have the potential of the PAR1 on it that it could be regulated through this receptor.
So, any more? Then I have one last question about the macrophages. Simply my interest. What do you think? Do you think that if you treat with rivaroxaban, you get less macrophage infiltration or more macrophage emigration from the plaque?
That would be interesting. I would be interested to measure the MCP1, which is responsible for the infiltration. And upon the literature that we know, there's not much literature about it, that there's reduced MCP1. So it's probably really the infiltration of macrophages in the plaque.
Okay. So, if there are no more questions, then I would thank the audience for the discussions, and I would like to thank the speakers for giving nice presentations that are in time, and the audience for the attention. And with this, I would like to close the session. Thank you very much.